Okay, we'll look at the section of ribosomes. So before we look at translation, let's look at the player, the organelle that catalyze translation, ribosomes, that will help us to understand the translation process next lesson better. Okay, so two types of ribosomes, right? The eukaryotes and the prokaryotes have different ribosomes. The prokaryotic ones is known as a 70S ribosomes, and the eukaryotic one is known as the 80S ribosomes. The prokaryotic 70S ribosomes made of two subunits, large subunit 50S and small subunit 30S. Eukaryote large subunit 60S, small subunit 40S. Okay, so to help you to understand, right, you can imagine that the S is kind of like size. Right, so it's called a sedimentary coefficient, but to explain it simply, it's kind of like mass or size. Okay, so the eukaryotic ribosomes are slightly larger, have slightly more mass. That's why it's of a higher sedimentary coefficient. So this is oversimplifying it, but if it helps you to understand it, then we'll just take it as size lah. Okay, so let's look at the structure, right, of each of the subunits. So we start with the large subunit first. Okay, so it is of course each subunit is composed of rRNAs and ribosomal proteins. Right, so each of the ribosomal subunits are a complex of nucleotides and proteins. All right, there are three binding sites for tRNA in the large subunits: the peptidyltransferase, the peptidyl tRNA site is in the center here, right? That is where the tRNA carrying the growing polypeptide will be. The amino acid tRNA site is here for the tRNA carrying the amino acid, and the exit site for the tRNA to be released. Okay, all this will come together when we look at the translation process. So for now, you just know that there are three different tRNA binding sites in the large subunit. Okay, in the large subunit, there's also an enzyme called peptidyltransferase that catalyze the formation of peptide bond. Right, so again, this will come into play when we look at the translation process. All right, for the small subunit, there is a mRNA binding site, right? So which will bind to the mRNA. Okay, so on the mRNA, right, in a prokaryotic mRNA, there's this sequence in the mRNA called the Shine-Delgado sequence that is before the start codon. Okay, so this Shine-Delgado sequence would bind to the mRNA binding site in the small subunit. For a eukaryotic mRNA, right, there's a 5' untranslated region, right? So in short, 5' UTR, that is also before the start codon. Right, so this 5' UTR would bind to the mRNA binding site in the small subunit. All right, then next, this one is not really protein synthesis, but I thought you should know, right, how new ribosomes are formed. Okay, so we call that in a eukaryote there's a nucleus, right, and within the nucleus there's a nucleolus which contains the genes that codes for rRNAs. Right, so the genes coding for RNAs, right, will be transcribed to form rRNAs. Right, so these are the RNAs. Right, these will remain in the nucleolus because rRNAs are not translated. Right? rRNAs remains as rRNAs, so they will remain in the nucleolus. Right, then there are genes outside the nucleolus in other parts of the nucleus that codes for proteins. Right, and some of these proteins are ribosomal proteins. Right, so these genes coding for ribosomal proteins will be transcribed into pre-mRNAs, and the pre-mRNAs will undergo post-transcriptional modification to form mature mRNAs. Right, so mature mRNAs would then leave the nucleus for translation in the cytoplasm, right, by existing ribosomes. Right, so these ribosomal proteins would be synthesized, and they would go back into the nucleus and into the nucleolus, right, to complex with the rRNAs to form the ribosomal subunits. The ribosomal subunits would then leave and go into the cytoplasm, and they will form the new ribosomes. All right, so this is a process. All right, I hope you understand. Okay, a little bit of the process of protein synthesis involved, of course, because of the ribosomal proteins, and also linking back to what you learned before, right? That ribosomes are formed in the nucleolus, and the nucleolus contain genes that codes for RNAs. Okay? All right, so that would be the section of ribosome.